My name is Wenyi Wang. I'm a professor in the Department of Bioinformatics and Computational Biology and Biostatistics at MD Anderson Cancer Center. So very nice to meet you all. It is my pleasure to be here and tell you about our development on computational deconvolution of cancer genomes and transcriptomes. So my lab's main research interest is to develop statistical methods and tools to study the evolution of human genomes and cancer genomes. And today's talk will be focused on the second part, the cancer genomes. As we all know that cancer cells continually evolve by acquiring genomic alterations, as is shown here, I'm trying to use the pointer here, that the different colors correspond to the cancer cells with distinct genetic alterations. And when selective pressure, such as treatment, is imposed, most of the cancer cells are killed, but unfortunately, a small group of cells, usually with a unique color, might escape that process and further expand and evolve. And that's why it is important for us to study cancer evolution. Related, just as humans, as many talks that you have heard so far, that the ecological context in this particular context is the transcriptomal profiles of cancer cells largely impacts its evolutionary trajectory. So I would like to start with a case study. So my group are studying androgen-indifferent prostate cancer in collaboration with Dr. Anna Aparicio at MD Anderson. So these are patients with prostate cancer that do not respond to androgen signaling inhibitors, which are the standard treatment for prostate cancer patients. And therefore, they have significantly reduced survival. So here we're studying two tumors that are derived from the same patient who is 1 year apart. These two tumors, as we see here, share the same TP53 mutation, suggesting that they share the common ancestor, cancer cell of origin. But at the same time, the first tumor presented androgen receptor negative, where the second tumor then transformed to an AR positive. The switch in AR status is a significant functional change in prostate cancer. So largely or broadly across cancers, the researchers nowadays are extremely interested in studying two spaces at once. The transcriptotype space, where multiple states of cancer cells exist, and then the genotype space. So our hypothesis is that understanding the trajectory of these changes at the same time will hopefully allow us to identify an approach or a way to enforce the cancer cells to a favorable states that will respond to treatment. The cancer cell's ability to respond or not respond to treatment, varying over time or space, whatever, had a traditional name called plasticity. And the plasticity has been closely linked with cancer cell stemness, a concept that was introduced more than 40 years ago. The plasticity in cancer cell stemness sort of hit a wall. It got the popularity, but went down because of all the controversial observations people have been observing. The single-cell sequencing technology came in and allowed the cancer researchers to be able to see these variations at the cellular level and realize it is no less than plasticity is no more than just changes in genomes and transcriptomes presented by the same group of cells. So our central hypothesis of this talk is that a fuzzy and evolving biological concept may be improved by mathematical definitions. So here I would like to introduce the huge iceberg, using iceberg as an analogy, that we would like to study the full spectrum, all aspects of an iceberg, even though the majority of the iceberg, the information is buried under the ocean and requires a huge amount of energy to understand it, such as using single-cell sequencing data to study patient tumor samples. So Anna and I, we spent many meetings trying to figure out a way to generate reliable single nuclei or single-cell RNA sequencing data from her clinical trial, here the Dynamo trial, but there are a lot of sampling and technical issues that we have to jump through. At the same time, the tip of the iceberg, or the bulk sequencing data, have been readily generated, are waiting to be linked to the clinical follow-up information. So our second hypothesis, so this is not a matter of finance, but a matter of how to generate reliable data. So our second hypothesis is that scanning through the tips of the iceberg across the ocean is informative and complements the investigating just a few icebergs in full length. One may ask, this is exactly what cancer researchers have been doing over the past 20 years with very limited findings. And that is because the accurate quantification has been a bottleneck, and that is due to the tumor heterogeneity. So here I'm showing you two zoom-in views of one H&E slide of a resected tumor sample used for pathologist report. So on the top view, you see try and use the pointer here. The cells, large cells with white space inside suggesting open chromatin states are the tumor cells, and these elongated, nice-looking cells are fibroblasts, and these smallish and dark, solidly stained cells are the lymphocytes. So on the top view, they're loose, sparsely distributed, but at the bottom view of the same slide, the lymphocytes are swarming and then seemingly attacking the tumor cells, and with the tumor cells in the neighborhood presenting less open chromatin state, suggesting a different cell state from the tumor cells in the top view. So to summarize, the sequencing data generated from bulk tumor samples are measuring a mixture of signals coming from different cell types and cell states, and the composition of these cell types and cell states vary largely across samples that are derived from the same or different patient. So our main goal is to use deconvolution models to help define features of plasticity. And here I introduce my definition of plasticity in the widest mathematical concept, which are changes in genomes or transcriptomes of the same groups of cells. So to define features of plasticity in human tissues in mathematical terms. So I introduced the transcriptome plasticity score measure for tumor cells and genetic diversity measure for tumor cells. We also have methods that can, the methods can be applied to immune cells, but I won't have time to talk about it in this talk. So transcriptomic plasticity. So the issue of tumor heterogeneity in gene expression that prevents the quantification of cell type specific plasticity is because simply the true tumor cell gene expressions are masked by the stromal cell expression, as is illustrated in this simple example where we thought we're comparing the red versus the green, so for example in D analysis, but we're actually comparing the orange versus the green because of the contamination of the stromal cells. And such superficial comparison then would lead to loss of missing the functionally important genes. All right, so methodology. So how do we resolve this issue? Experimentally, there's laser capture microdissection, which requires the pathologist and very fancy microscope, not the usual microscope. And so the pathologist will need to use the microscope to tease out the tumor cells and stromal cells. It's laborious, and it does not scale up to a regular biomarker study with, say, 200 patient samples. So in silico approach seems to be the major way that we can break through and resolve this issue. So there have been many methods that have been developed for deconvolution, and simply most of them follow this linear equation, A times X equals to B. So here A is a matrix of pure cell expressions, so say 10,000 genes, and then with 3 mixed pure cell type component, so 10,000 by 3. And then X is a vector proportions, a vector of 3. A times X then gives the B, which is the observed heterogeneous expression data, a vector of 10,000 in my example. Earlier deconvolution methods tend to say, oh, let's assume that we can get X, which is the mixing proportion, and then solve for A. But it turns out X is actually the most difficult estimate. That is just, there's no reliable way that can give us a good starting point for X. So alternatively, more recent methods all more or less rely on the knowledge on A for some of the genes. So as you can see that we don't really need 10,000 genes to be able to solve for X. We just need, say, 50 to 200, etc. So these are the genes, however, that we need to know the exact expression values for each of the mixing component. And then they're called reference or marker genes. And then once we have these genes, then we can recover X by supervised approach, just like linear regression. So this approach applies well to, say, deconvolve immune cell types because of the existence of marker genes, therefore cybersort and timer, etc. However, when we try to separate tumor cells versus others, we just can't possibly find, I can't possibly find marker genes or a set of marker genes that's reliable to represent the tumor component. As we aim to understand the tumor plasticity, so our goal here is to perform a semi-supervised deconvolution here I'm using a two-component to illustrate the point. So here we set up as in our input data is first the mixed expressions, as you see on the right. These are a heat map, simply gene expression, genes on the rows, and then samples on the columns, and as well as an additional set of samples that measures expression for one of the two mixing components. So here I'm actually showing you an in silico mixed experiment generated gene expression data where they're mixing RNAs extracted from lung and then brain tissues from rats. So then where output is, we want to estimate the mixing proportions pi. We don't believe that we can obtain it just by other approach. And we would like to recover the cleaned up full matrices for each genes and each samples for tissue, the mixing tissue component 1 and not shown here, the mixing tissue component 2. So this is quite an ambiguous goal. So how do we solve it? So our model is quite simple and straightforward. It's a linear constraint. And then here we have Yig for gene G and sample I. Yig equals to pi I times Tig plus 1 minus pi I times Nig. Pi is the tumor proportion for sample I, and Y is the observed expression, T is the tumor component, and N is the non-tumor component. So non-tumor component includes stromal cells, immune cells, etc. I'll call it normal, and since it's N from now on, but it's nothing normal because these are the cells existing in the tumor tissue. It just, except they're not cancer cells. So first of all, this linear constraint, although simple to write, it's not simple to solve. This is not a finite mixture model, but a linear combination of distributions where the mixing proportion changes over samples. All right, so here I'm introducing a toy example just to hammer, to kind of help you understand the two main challenges here is the model identifiability and high dimensionality. Let's dumb it down a little. So first, let's say N is known with minimal variance. So no variance. Then we have Y star Ig equals to Yig minus Nig. Then for each gene G, we have Yig star equals to pi I times Tg minus Ng. So here we have n plus 1 parameters to estimate, but only n observations, so unidentifiable. And similarly, for each sample I, we have Ig star equals to pi I times Tig minus Nig. G plus 1 parameters to estimate, only G observations, again unidentifiable. Of course, you would say, one would say that when pi's are variable and t, there are differences in pi's and t's, then we can borrow strengths across multiple samples and genes and then perform a matrix decomposition. But that can be performed when we assume no variance. In reality, in the real data, it's capturing the variance is what makes this problem difficult. And then high dimensionality. So the total number of parameters to be estimated, there are two steps. At the first step, we estimate the mixing proportions corresponding to the number of samples and the mean and variance for the distribution, gene expression distribution for each gene. So it's the number of genes times 2. So for example, a regular biomarker study is 200 plus 20,000 times 2. And once that's given, then the second step is to recover the full individual deconvolved matrices for each of the component. And that's 20,000 times 200 times 2 values that we will need to estimate and then be able to complete the job. So how do we resolve this? So we have so far moving forward pretty quickly. so we came up with a solution to perform deconvolution for both the two-component mixture model, first the two-component, and then the three-component mixture model, where the goal was to separate the non-tumor component into stroma and immune, because we were particularly interested in immune proportion changes across tumor samples. the few methodological notes is that we decided to use the log normal distribution as it turns out to fit our model assumption the best, which then led to a kind of numerical issue that we continue to struggle with until today, as in the full likelihood for the 2-component has 1 integration, which is okay to deal with, and then 3-component mixture has double integration, which then lead to my next slide. Actually, so far we have our main solution and the algorithm behind the DMXT was introduced by my former PhD student Wenyi Wang when he was working on a 3-component mixture model. And then here's the method. So we decided to first understand that the space of the parameters to separate them into the genome-wise parameters and sample-wise parameters, as is shown here in the bipartite plot. So it turns out they have, well, as we observe, we see that they have this conditional independent relationship, which then allows us to use an iterated conditional modes approach, which iteratively maximize likelihood of each pair of the genome-wise and sample-wise parameters conditioning on each other. So we came up with this method and then found that it was already introduced into literature, early on for solving imaging data problems, and has been proven already that its likelihood never decreases and is guaranteed convergence to local maximum, which is great. However, as with this picture that I just took on my flight to LA, that we are not satisfied with local maximum. And as I alluded to early on, is that our likelihood space, because of the high dimension curves of dimensionality and identifiability, is just local maximum is not good enough. We have to find the global maximum. So long story short, without describing the details, we came up with the gene set-based component merging with the goal of locating ourselves around the global maximum when we start the ICM search. All right, so with all of these methods development, we then went on and then performed various validation studies to benchmark our approach. And here I just mentioned one study, which is the laser capture microdissection study performed by our collaborator and Dana-Farber, the pathologist. So here they took the 23 mixed tumor samples from prostate cancer and then manually dissected them out for the tumor cell and then stroma cells and kept the remaining mixed tumor tissues and then sent all three parts for expression profiling. We then took the mixed tissue versus the pure tumor tissue and then round DMXT, and then the mixed tissue and the stroma component and round DMXT again. Hence the pi T here and then pi S, which are independent rounds. So first, when we compare the pi T hat with the pathologist's visual inspection of cellularity, we saw absolutely no correlation. We then ask, okay, so maybe just maybe that we're measuring something that the pathology is not seeing because we're looking at the molecules and pathology is seeing something else. So we then say, how about we compare the pi T versus pi S? If our method works, then they should sum up to 1. So as you can see here, pi T versus 1 minus pi S lies nicely on the diagonal line. Therefore, confirming that we are estimating a proportion, just it's different from the pathologist estimate. We then performed the 3-component deconvolution by in silico, through collaboration with biologists, by in silico mixing the lung cancer cell lines, fibroblast, and immune cells. And then in that mixing experiments, we calculated both the cell proportions for the 3-component and the transcript or the RNA amount proportions for the 3-component. And turns out our DMXT estimate is most consistent, is highly consistent with the RNA proportions, but not the cell count proportions. And why? That's because different cell lines, as many of the single cells researchers here know very well, different types of cells contain a completely different amount of RNAs. So we should have known this when we developed the model, but we actually found it through experimental validation. So why is this then important biologically? Because just as DNA content, some of you know the ploidy, the RNA content was also of interest for over the past many years as you see here, 1979, as old as me, and I'm telling you how old I am. In order to differentiate atypical hyperplasia from truly malignant cells, the main limitation is that in the past, biologists are limited to low throughput methods as limited by the number of tumor cells they evaluate or the number of genes they evaluate. And then the single-cell RNA sequencing came in and allows for large-scale quantification of cells. And this is first the Caltech's group, shortly after the invention of single-cell sequencing, showed that there are significant variation in total mRNA content across cells using spiking quantifications. And then the statisticians came in and say, well, there's definitely a transcriptome size effect together with the library size effect, and then we should remove these sizes before the differential expression analysis. Early 2020, a science paper showed that these are the Stanford researchers evaluated more than 20 single-cell data sets and showed that single-cell transcriptional diversity, as measured by simply the total number of expressed genes, a surrogate for total amount of RNA, is a better hallmark of developmental potential than any specific gene pathways alone. So what does that indicate to us as we found with DMXT? So far there's no systematic study about the effects of total mRNA expression levels in tumor cells in human tissues at scale. And to prove that it's actually a feature of cancer cell plasticity. So our study aims to fill in this gap in knowledge, and this is a paper that just came out at Nature Biotechnology. So we started out by first checking, okay, is there high diversity in tumor cells in our data? So we obtained the single-cell RNA sequencing data from 10 patients in 4 cancer types: colorectal, liver, lung, and pancreatic. And 2 of the 4 are local from our collaborators, and therefore we have immediate access to the patient clinical follow-up. And then we basically, every month, we ask them for an update. First is yes, we do observe much higher diversity in the total mRNA expression, as we see here in both the gene counts and total UMI counts in the tumor cells versus the non-tumor cells. We then look further into, say, colorectal cancer. There are 2 patients. Patient 1 relapsed at 4 months, and patient 2 remained relapse-free at 16 months. We found that indeed the Seurat clusters, there are multiple Seurat clusters, and then that present distinct gene counts as well as the total UMI counts across the tumor cells. And with the cluster on the far right presenting the highest amount of total UMI compared to any other clusters within tumor cells or across other cell types. And this is only presented in patient 1, which relapsed in 4 months. And further looking into the biological activity within the high UMI cancer cells, we found that it's not cell cycling or proliferating faster than other cells, but it is enriched for genes participating in epithelial mesenchymal transition, which is the best known feature for cancer cell plasticity in the literature, and it is indeed enriched for stemless gene signature gene sets, therefore suggesting a stem-like cell state. We repeated our finding in that higher diversity in total mRNA expression in tumor cells is observed from patients with poor prognosis in the other 3 cancer types. But as you can see, we are very limited with very small number of patients. And the reviewers asked, the editors asked, we have looked into literature, we cannot find more. The reason why is that when the single cell studies are published, they are not waiting for the 5-year or 10-year follow-up for that same patient. So if you're looking at 2018 and onwards publication, there's no follow-up information that's provided. All right, so we therefore hypothesized that a quantification of, got too excited. So we therefore ask, okay, how about is this signal observed at the bulk level? So we took all the single cells and calculated the mean UMI counts across all tumor cells divided by the mean UMI counts across all non-tumor cells, and saw that the clear difference across patients at the pseudo bulk level. We therefore hypothesized that a quantification of average tumor cell total mRNA levels in bulk samples across large patient cohorts is useful to track tumor phenotype and its direct link with clinical outcomes. So why haven't people done it? There are two challenges that prevented the previous studies from addressing this question. So back in the macroarray era, the Richard Young's group has already nicely summarized the first issue as in when individual genes are increased in expression, then after normalization, such effect remains. But if all genes are increased in expression, then after normalization, this effect is removed together with the normalization. So it's a perfect confounding situation. So he already complained about that. And then the other challenge is, of course, the tumor heterogeneity, as we're interested in tumor cell activity, but we're measuring a mixture of signals. So our solution is to use one challenge, deconvolutions, to solve the other, because the deconvolution contrasts the two groups of cells within the exact same experimental condition, hence canceling out only the technical effect by default. So we therefore introduced tumor-specific total mRNA expression, TMS, which is the total mRNA expression per haploid genome for the tumor cells divided by the non-tumor cells. So there's three main main steps. The first is, as I mentioned, in order to cancel the technical effects, we take the ratio of the tumor cells versus non-tumor cells. And this ratio is equivalent of an odds of the transcript proportions that we can estimate, pi over 1 minus pi. And then we further adjust for the number of cells, which is not observed but can be calculated as the odds of the rho, which is the tumor cell proportion. We then further adjusted the number of chromosomes or ploidy because we're less interested in the dosage effect, but more interested in the fundamental biological feature. Therefore, TMS becomes an odds ratio of the two proportions and then further adjusted by ploidy. So with a tumor sample, we need matched DNA-seq and RNA-seq data. We can then calculate TMS by first calculating the tumor cell ploidy and tumor cell proportion. As some of you know, these two values are not the easiest to estimate. They're an identifiability issue, and therefore with TCGA we took the consensus between the two methods, ASCAT and absolute, and turns out we only needed to drop 5% of the samples, which is great. And then with RNA sequencing data, we use DMXT to estimate the tumor-specific total mRNA proportion pi. So we realized that gene selection is essential, as well if we include genes that do not follow this nice bimodal, the single mode for the normal component, single mode for the tumor component, for example, multimodal in the tumor or large variance in the both component, then including these genes will introduce large biases in the pi estimation, which here we are really going for the accurate estimation. So we introduce a profile likelihood approach, and because the N and T are not observed, neither is observed, but profile likelihood allows us to identify genes that is most likely to follow the underlying distribution the best. So the gene selection score, correspondingly, the smaller the gene selection score, then the better the gene is more likely to follow this desired underlying distribution. With TCGA data, for each cancer type, we ended up selecting the top 1,500 to 2,000 genes with the small gene selection scores. So with all of this effort, we then calculated TMS for 6,580 patients in the TCGA, ICGC, Metabreak, and TracerX studies. And here is a landscape distribution of TMS. And we also further did a benchmarking study where we mixed the cell lines and have the true TMS values and be able to evaluate the uncertainty in the TMS estimation. So what you see first is that the variation across patients is much larger than the variation we will observe when there's one TMS, a true TMS value. And second is that the ranking of the TMS is meaningful in that more indolent cancer types tend to present low TMS values and more aggressive cancer types present higher TMS values. We therefore hypothesized that TMS is associated with clinically useful prognostic features, characteristics. And so here I'm showing you the HPV status in head and neck, triple negative HER2 ER/PR status, and PAM50 status for breast cancer, Gleason score for prostate cancer, luminobasal for bladder, and the type 1/2 for renal papillary. So of all of these scenarios that we investigated, the higher value of TMS is significantly associated with a feature or a subtype presenting the least favorable prognosis. With breast cancer, we then accessed the MetaBreak and was able to replicate our observation in TMS using an additional 1,500 patients. So this is probably the most striking observation, as we found that TMS further refines prognostication on top of pathological stages, which are currently used in clinic for decision-making in all 13 cancer types in TCGA where the stage information is available. As you show here, they're all significant either in the black, the main effect of TMS, or the green, the interaction between TMS and stage. The only thing that was not significant is ER-positive breast cancer. And then by further looking at the MetaBreak, we found out why. Is because in two reasons. First is the follow-up of breast cancer is limited to 5 years in TCGA, as compared to a MetaBreak is 20 years. So by the way, 20 years ago, there was no single-cell data. And then second is ER-positive has a perfect mixture of both breast cancer patients sent for chemo as well as those that are not sent for chemo. And as we found that for cancer types that mostly have larger patient population without chemo, we observe a positive effect, so high TMS, worse prognosis. But with breast cancer, for example, triple-negative breast cancer, where most patients are sent for chemotherapy, we found patients with high TMS presenting a better prognosis. And similarly, with ER-positive and HER2-negative, which is the largest breast cancer population, these are the patients that are currently relying on Oncotype DX, a 16-gene signature panel, to advise whether these patients should go for chemotherapy or not. And then the high-risk group are those that will be advised for chemo. And we found that a subset of these patients with low TMS may not benefit from the chemotherapy. So to summarize, the TMS has the potential to serve as a marker for worse prognosis as well as for better response to chemotherapy across cancers. And moving from intra-tumor heterogeneity to intra-tumor heterogeneity, we calculated TMS from TRACER-X study, a multi-region profiling with 3 to 5 regions per patient, and we calculated TMS for each patient. What we found is TMS, again, a transcriptome plasticity score, is significantly correlated with % subclonal copy number aberration, which corresponds to ongoing chromosomal instability, where it has nothing to do with transcriptome, at the regional, across all regions, across all samples, as well as at the patient level, when we summarize across regions to TMS max. And TMS max can be further used to dichotomize these early stage lung cancer patients in prognosis. So regional TMS identifies spatial heterogeneity in transcriptome dysregulation and provides prognosis from subpopulations of tumor cells presented in one region. In summary, tumor cell total mRNA expression, or transcriptome ploidy, in analogy to genome ploidy, provides a key feature to track tumor cell phenotypes. So next, the genetic diversity. So my group has been part of the PCOG working group and timing and intra-tumor heterogeneity of 2,600 cancers across 38 cancer types over the past 7 years. So we were focused on developing statistical methods for the accurate measurement of high-throughput sequencing data. So we developed Muse for subclonal somatic mutation calling, and then CLIP for subclonal reconstruction, which I'll talk a little bit more in the next few slides, and also Caesar for consensus subclonal reconstruction. So our goal here is to identify subclones of cells through mutational events as a first step towards studying cancer evolution. So here on the left is an example that we start with cancer cell population A with some mutations, and then it divides into two groups. Population B acquire new chromosomal, an additional chromosome, and population E acquiring additional mutations. And then B diverge into C and D, which acquire additional unique mutations. And there's usually, when we profile these tumors, there's usually contamination of stromal cells, where we assume there's no somatic mutations and the diploid genome. So in this context, what we're focused on is not building the tree yet, but we're focused on a more earlier task, which is to classify mutations correctly. So here I'm using the illustration on the right to describe what we are working on. So we start with, again, a cancer cell population with green mutations, and then we calculate its cellular prevalence, which is equal to 0.9. Why 0.9? The prevalence, because there are 10% contamination from the stromal cells. And then these cancer cells further evolve by 2/3 of them acquiring additional mutation in blue. This is a very simplified example. So then the blue mutations correspond to a cellular prevalence of 0.68. And then furthermore, among these cancer cells, 2/3 of them acquire the additional brown mutation. Therefore, for the brown mutations, the cellular prevalence is 0.47. So our goal is, when we're given all the mutation cause and VAF, we try to adjust for tumor purity and copy number aberrations in order to cluster the mutations, assign colors to all of the mutations, and their shared cellular prevalence. So that's what we're trying to do. I'm not gonna. So there are many methods that have already been published. I'm just focused on what we are trying to add to the literature, which is to improve the estimation. The popular methods currently available is our Bayesian methods using derivative prior. So basically, we're running one MCMC for each sample, and then that requires the post-processing a lot of manual tuning, and not to mention it takes a long time to run an MCMC. So we would like to develop a completely orthogonal estimation approach to DP for subclonal reconstruction in order to provide a robust consensus outcome. And then we chose the likelihood model with regularization, and specifically our current penalty is based on the SCAT soft clip absolute distance penalty. Of course, adding the SCAD to the subclonal reconstruction model requires a lot of mathematical derivations, which is available online in our bioarchive. So here I just focus on the conceptual conceptual advancement. So we follow this JASA paper, use a homogeneity pursuit framework, in the sense that previously the penalized regression likelihood focused on sparsity, as assuming we only have a small fraction of coefficients are non-zero. But here we're focused on homogeneity, as all of the coefficients are non-zero, but they are similar, so they can put into just a few groups. So we're looking for homogeneity. And this is a particular specific function that we try to maximize within our model, and with the lambda, the hyperparameters that are set at these 11 values. So, and these are the homogeneity pursuit plots, we call it, for two real data, where we start out when the lambda, the sparsity, the homogeneity penalty is low, then we have many clusters of cellular prevalence, and then when we increase the penalty, then we converge to 3 clusters in both examples. At last, we introduced our method CLIP, clonal structure identification using pairwise penalization using SCAT penalty. So here we're showing you the results on the computational time for CLIP. It takes a CLIP less than a day to finish running the entire PCOG, and just a few hours to finish running the entire TCGA data. And then specifically for a sample with 5,000 mutations, CLIP finished running in 1 minute, as compared to the variational inference method 4 minutes, and the Philo WGS 1 day. All right, so and is it accurate? It's fast. Is it accurate? So we compared using 1,500 peacock samples, compared CLIP with the consensus, and then found that actually the highest consistency between CLIP result across as compared to 10 other methods versus the consensus, supporting that it is accurate. So now, with all the time that we got saved by running CLIP, we started to ask what are the follow-up interesting questions. So first, we looked at the 588 samples where matched whole genome sequencing and whole exome sequencing data exist. And we identified the mutations that are shared by these two platforms and ask whether their clonal versus subclonal assignment by CLIP have switched or not. And we used an inter-rater agreement measure and found that across 588 samples, which come from 21 cancer types, the consistency is largely high. So the median consistency value is 0.81, corresponding to a perfect agreement. We then calculated a coding sequence subclonal fraction, which is basically the subclonal CNVs out of all of the coding SNVs. So what we found when we compared now all of the samples in Peacock 1900 versus 7700 in TCGA, we found this amazing consistency in the ranking of the median subclonal fraction of the cancer types, with cage, thyroid, prostate, the indolent cancer types nicely located on the far left, and then lung and bladder cancer, and then skin and melanoma nicely located on the far right. And we believe that this is suggesting further, first, that our algorithm works, and second, the subclonal fraction is a biologically relevant and robust feature for cancer. So last, I'll end with closing the loop that with all of these deconvolution methods that we aim on efficiency and accuracy, now we have all the metrics that allow us to then ask how are transcriptomic plasticity and genomic diversity acting together and then in order to identify a better target for cancer treatment. To conclude, so we have developed deconvolution models to help define features of tumor plasticity in mathematical terms in order to improve the genomic and transcriptomic definition for heterogeneous tumors. And all of methods that I mentioned today are freely available online. And this is a huge team effort. And the work that I present today, most of the lab members that produce the work are here. So please feel free to talk to them. Shuangxi, Ji, and Jeffrey Jiang, and Matthew Montierth, Shuangxi, and Yujie. And I'd like to thank all of my collaborators, a huge group of physician scientists, cancer biologists, and computational biologists supporting us, the project with their expertise. And I'll end here. Thank you.